In this chapter, we'll cover microbial metabolism. So metabolism is a balance between all of the chemical reactions in your body, and we either break things down or we build things up. And basically the biochemical pathways that we talk about, you have a starting point, then some kind of intermediate, and then an end product. And enzymes play a very important role in our biochemical pathways. So we have three categories. We have catabolic pathways. That's when you will break down substances and release energy. So you go from large to small. Anabolic pathways, where you will actually use energy and take smaller molecules to make bigger molecules. And sometimes we have what are known as amphibolic pathways, and that's when you can actually both break down and build up again. ATP, that's our energy, and I'm sure in other classes you've covered ATP. And some important things with ATP is that by removing the terminal phosphate group from ATP by what's called dephosphorylation, you release energy and adenosine diphosphate, or ADP. Now, when the cell needs more ATP, it can add a phosphate group to the ADP via a process called phosphorylation. So enzymes are important, and they are protein catalysts that help reactions occur under cellular conditions. So they will actually speed up a reaction. And these are just some general characteristics then of enzymes that we have. So basically what will happen is a substrate will bind to the active site of an enzyme, and then the enzyme will react or work on that substrate, and in this case you'll get two products at the end. So you're breaking down one big compound into two smaller ones, and then the enzyme is available again to continue the cycle. So they're not used up in reactions. So please know that the site where a substrate binds the enzyme is called the active site. Now we have different categories of enzymes, and an enzyme that doesn't have cofactors is called an apoenzyme, or an inactive form. And an enzyme that has cofactors is called a holoenzyme, or active form. So in this case, the cofactor is here, so that would be an active form. Down here, this would be your apoenzyme. We don't have our cofactor, so it's not active. So certain coenzymes will act as electron carriers, and sometimes they're called electron taxicabs, electron shuttles, but they pick up electrons from one reaction and then move them to another compound in that reaction, or to another reaction altogether. So NAD+, NADP+, FMN, and FAD are good examples of coenzymes that are cofactors. Now, effects of temperature. If you lower a temperature, you slow the metabolism down, so enzyme activity goes slower. Warmer temperatures can increase the enzyme activity to a point. However, if you go too high, you can denature the enzyme. Okay? So you can denature an enzyme by going too high in your reaction. Now pH again, above and below, you're going to disrupt the bonds. So extreme changes in pH can also lead to protein denaturation or enzyme denaturation. Now, a lot of medication out there is actually based on inhibiting enzymes. So please know competitive and non-competitive inhibition. A competitive inhibitor binds the active site of the enzyme and blocks substrate from binding. So that's competitive inhibitor. Binds the active site of the enzyme, and that will block the substrate from binding. Now, a non-competitive inhibitor, non-competitive, binds the regulatory site of an enzyme that can change the shape then of the active site of the enzyme, and then the substrate cannot bind. Also important in making ATP is what's known as redox reactions, and that's oxidation and reduction. So please know that oxidation is a loss of electrons and reduction is a gain of electrons, and a lot of times that's coupled together. So, as I mentioned before, certain cofactors can actually be electron shuttles. So the two I want you to know for your exam would be NAD plus and FAD. They can act as electron shuttles. So they can take on the electron and then the electron gets passed along, etc. in pathways. So we're going to go through and I'm going to tell you what I'd like you to know about glycolysis, Krebs, and electron transport. So for glycolysis, please know your total energy yield is 2 ATP and 2 NADH. Know the 3 steps. Glucose is converted to fructose 1,6-diphosphate. 2 ATPs are used in this step. In the second step, fructose 1,6-diphosphate is broken down into 2 3-carbon molecules. Between 2 and 3, there's several steps involved in there, but please know that the 2 3-carbon molecules are converted to pyruvate. In this step, 4 ATPs and 2 NADHs are generated. Now, an important fact is that between glycolysis and the Krebs cycle, we need to convert pyruvate into acetyl-CoA. So please know that pyruvate will be converted to acetyl-CoA, and this is considered the intermediate step then between glycolysis and the Krebs cycle. So please take a screenshot of this and upload it to your Dropbox. So for the Krebs cycle, all you need to know is the following. The energy yield is 2 ATPs, 6 NADH, and 2 FADH2. In our first step, you're going to take a 4-carbon compound called oxaloacetate and combine it with the acetyl-CoA, which has 2 carbons, and you make citrate, which is 6 carbons. Now, there's a series of steps then that are going to allow you to go back. So again, this is a cycle, and citrate is converted back to the oxaloacetate, and in that process, 2 carbon dioxide are lost. Electron transport, wow, that looks pretty complicated, but I'm going to break this down. So for your exam, you'll have to know the steps of electron transport. First step, NADH or FADH2 will come in from glycolysis or the Krebs cycle. So keep in mind in bacteria this happens at the plasma membrane. Some of this happens in our cytosol and some happens in our mitochondria. Hydrogens are then removed and pumped across the membrane from a low to a high concentration. At the same time, the electrons will be passed along carrier molecules in the membrane. Once you get 6, and that, guys, varies depending on whether you start with NADH or FADH2. Could be 6, could be 10. But we're going to say, for testing purposes, once 6 hydrogens accumulate, they come back across the membrane through ATP synthase. And if you start with NADH, you will generate 3 ATP. And if you start with FADH2, you will generate 2 ATP. And please remember, amphibolic pathways simultaneously function in catabolism and anabolism. Now, these are just a few tests. I'm going to go through them a little bit quickly. These are ones you'll use in the laboratory to identify an unknown. You can test for amino acid catabolism by looking for deaminases, decarboxylases. We can also look for sulfur production. Here the black indicates that sulfur is, so these are some of the media you could use for that. Fermentation, we're going to be using phenol red as a pH indicator. If it turns yellow, that shows that pH is lowered. And this Durham tube here, if you see that little bubble or gas, that means gas is also produced with that. MRVP, again, the MR is looking for mixed acid fermentation. VP is going to detect acetone. Again, those of you in the lab, this is going to be important to you as well because you'll need this information. Oxidase test, a beautiful blue color. And for catalase, bubbles would be positive if that enzyme is produced in this experiment.